This is the lecture for Chapter 11, Mendelian Patterns of Inheritance. Genetics is the area of biology that studies how information for particular traits is transmitted to offspring. And it also involves studying how that information is expressed in the following generations. And often you can see this in a family photo if you have a multi-generational family photo. So you have the grandparents, and the grandparents had particular traits. They transmitted the information for those traits to their children. And then, of course, that information was then transmitted to grandchildren. And often in a family photo, you can see similarities in characteristics in all three of the generations. While it's interesting to look at family photos and observe which traits are transmitted from generation to generation, genetics is very important for agriculture. Agriculture is the science of cultivating plants and animals to produce food and other products. As long as there has been agriculture, people have been interested in how advantageous traits are transmitted from parents to offspring. So, for instance, a farmer may observe that there's a particular corn plant that grows very well in hot, dry climates. And then that same farmer may have observed how another corn plant seemed to produce twice as much corn as normal corn plants. And then what that farmer would want to do is cross those two plants, breed those two plants together, and produce an offspring plant that ideally would be able to survive well in hot, dry summers and produce twice as much corn as normal corn plants. And that is the practice of selective breeding, taking two parents that have advantageous traits and breeding them together to hopefully develop a new strain of more productive and more resistant plants and animals. Another example of selective breeding can be seen in all the different breeds of dogs that we have. Dogs were first domesticated about 20,000 years ago, and through selective breeding, we now have about 400 different breeds of dogs that are officially recognized. And those dogs, over time, have been bred for many different purposes, some for hunting, some for herding, sometimes they were bred for color, sometimes they were bred for size. But the end result is that we have many different types of dogs with many different types of looks. So when you look at a Chihuahua, that is a very different dog compared to a Bull Mastiff. And one more modern example of dog breeding is when you take a Labrador and a Poodle and you breed them together, and the offspring is a Labradoodle, which is very popular nowadays. Now, when selective breeding was performed in agriculture or in dog breeding, many people observed that it looked as if there was a blending of the traits, that you take the two parents, and if you blend their traits together, that you get the offspring. But if that was true, if that is the mode of genetic inheritance, then over time you would actually lose variability. So if every time the genetics, the genes, the traits blended together in the offspring, then over the generations you would have less variability, and that is not the case. Mendel is known as the father of genetics, and Mendel was an Austrian monk who lived in the late 1800s. And not only was he a biologist, but he was also a mathematician. And he was very interested in the question of how traits are transmitted from parent to offspring. And in order to investigate this question, he decided to use pea plants. And a couple of reasons he decided on the pea plants were because, first, pea plants are very easy to grow. And secondly, they have a short generation time. So he would be able to generate a lot of data in a relatively short time. And another thing about the pea plants is that they have objective characteristics. So some of the characteristics he investigated were seed shape, seed color, flower color, pod shape, pod color, flower position, and then stem height. And these were characteristics that were objective. He didn't need to interpret them. Anyone who looked at the plants would be able to evaluate the same data. And the last important thing about the pea plants is that he could generate true breeding plants. So by allowing a plant with particular characteristics to self-pollinate after a couple of generations, all of the offspring from that self-pollinating plant would be identical to the parent. And that's what we call a true breeding plant. The first thing Mendel wanted to investigate was the blending concept of inheritance. And the idea of the blending concept of inheritance is that the traits in the two parents are blended together to create what you see in the offspring. So to do this, Mendel started with a true breeding short plant, and he crossed that plant with a true breeding tall plant. And if the blending concept of inheritance is true, then all of the offspring should have a medium height. So when he actually performed this experiment, what he observed is that there were no medium pea plants. So none of the offspring had a medium height. And so this indicated to Mendel that the blending concept of inheritance was not the correct method of inheritance. After almost a decade of performing experiments and analyzing over 10,000 individual pea plants, Mendel came up with 4 basic conclusions. The first conclusion we call the particulate theory of inheritance. And this basically states that parents transmit discrete particles to their offspring, and those particles determine the characteristics of a trait. And sometimes those particles are expressed and sometimes they are not. Now, we refer to this as a theory of inheritance. Theory is something that is accepted as true because now with modern genetics, we know that those particles that are being transmitted from parent to offspring are genes, which are segments of DNA. But of course, Mendel in the late 1800s, they had no concept of genes at that time, but he did realize that there were particles that were transmitted, particles of information that were transmitted from parent to offspring, and they were not changed in the process. The second conclusion he made is the law of dominance, and this refers to the particles. So he observed that some particles for a trait are dominant over others. So when he says dominance, that means some of the particles, now we know them as genes, are more likely to be expressed than other particles or other genes. The law of segregation refers to how those particles are separated during gamete formation, and I'll talk about that in a little bit. And the last law he identified is the law of independent assortment, and this refers to when you are comparing 2 traits, how the transmission of 2 traits from parent to offspring, and I'll also discuss that later in the lecture. At this point in the lecture, it's important to go over some basic vocabulary words. And the first word is allele. And an allele is an alternate form of a trait. And to Mendel, the allele was that particle of information that is transmitted from parent to offspring. And so now, of course, we know that an allele is a gene. So for each trait, Mendel observed that there are different alleles. So if you're looking at the trait of height, he noticed there were tall alleles and short alleles. The next 2 terms are dominant and recessive, and these describe the actual alleles. If an allele is dominant, that means you need only one allele to get the trait. And for height, the tall allele is dominant. And it's dominant, so we write it with a capital letter. A recessive trait is one where you need 2 of the alleles to get the actual trait. And with height, short is recessive. And when you're writing it out, you use the same letter, but use a lowercase to indicate it is recessive. So dominant alleles, you need only one allele to get the trait. Recessive alleles, you have to have two alleles in order to get that trait. The next two terms are homozygous and heterozygous, and this refers to the alleles that you find in an individual. And Mendel recognized that each parent contributes one allele to the offspring for each trait. So if we're talking about height, every offspring from two parents has two alleles for height. And if that individual is homozygous, that means they have the same allele for the trait. So a homozygous dominant could have two capital T's, two tall alleles, or you could have a homozygous recessive with two small alleles, so two lowercase t's. So that's homozygous. Heterozygous means the individual has two different alleles. And so to be heterozygous for height would be one tall allele and one short allele. Then two more terms, genotype and phenotype. So genotype refers to the actual alleles that the individual has. So the two alleles they have. Are they homozygous dominant, homozygous recessive, or heterozygous? That would be the genotype. The phenotype is the physical expression. So what you actually observe. So the phenotype would be if the plant is actually tall or short. So genotype is the genetic makeup, phenotype is the physical observation. Then the last couple terms that you should know relate to the generations, and this is involved in the crosses. The P generation, this refers to the parents, so those are the original parents. Then F stands for filial, and filial means son or it means child. So the F1 generation are the children from the P generation. And the F2 generation are the children, the offspring from the F1 generation. So if you're talking about humans, the P generation would be the parents, the F1 generation would be their children, and then the F2 generation would be their grandchildren. So 3 generations. Punnett squares are closely associated with Mendelian genetics, and a Punnett square is a tool that we use to predict the probability of genotypes from two particular parents. And I just want to point out that Punnett squares were not used by Mendel. They were developed much later in the early 1900s by a professor named Reginald Punnett. So he was teaching his students about Mendel's results, and he came up with this process, a tool, basically to make it easier to predict the probability of genotypes from a particular cross. And in this lecture, we will be using Punnett squares to analyze some of Mendel's observations. So first, when doing a Punnett square, the very first thing you need to do is to determine the genotypes of the parents. So when you are given two particular parents, you need to figure out what their genotypes are. Remember, the genotypes are the two alleles they have for a particular trait. Once you have determined that, then you need to determine what types of gametes are produced by each of the parents. So how those two alleles will segregate into the different gametes. Once you have determined that, then it's very easy. You just fill out the Punnett square and then calculate the probabilities of the genotypes. And once you know the genotypes, you can figure out the probability of the phenotypes. Now I'm going to show you how to do a Punnett square on a basic monohybrid cross. So mono means one, so a monohybrid cross is looking at the inheritance of one trait. And then in this example, the trait we are looking at is height. So the two parents, one is a tall parent, the other is a short parent. Now the very first thing when you are performing a Punnett square is to determine the genotype of the two parents. So tall and short or dwarf, those are phenotypes. So the very first thing you have to do is determine the genotypes. And in this case, the genotypes are given to you. So the tall parent's genotype is homozygous dominant. The dwarf or the short parent's genotype is homozygous recessive. So we have the genotypes right there. Second thing that needs to be done for a Punnett square is to identify the two types of gametes that each parent can produce. Now remember with gametes, when I talked about meiosis, gametes are haploid. That means they have one copy of each chromosome or each gene in them. So when this tall parent is producing gametes, half of the gametes are going to have this tall allele, and the other half of the gametes are going to have this tall allele. And for the short plant, half of the gametes are going to have that short allele, and the other half of the gametes are going to have this short allele. All right, so that's easy to figure out the gametes for those two parents. So the next step, the third step, is to set up the Punnett square. So the Punnett square is a square, and for a monohybrid cross, it's going to be a 4 by 4. So the tall parent will be here, and the short parent will go up here. So for the tall parent, we put the two types of gametes the tall parent can produce here in this column. And then the two types of gametes that the short parent can produce are put up here. And then we just fill it out. So by convention, if there is a dominant allele, that goes first. So we're going to cross this row first and across the bottom row. And the recessive alleles will fill in the columns here, and the column here, and here. All right, so these are all the probable or possible genetic combinations for the offspring for these particular parents, for a homozygous dominant and a heterozygous recessive. So now, after we've completed the Punnett square, we analyze the results. So there are 4 possible results. So each quadrant is worth 25%, but if you notice, they are all the same. So for genotypes, 100% of the offspring will be heterozygous. And if you're analyzing for phenotypes, you know that tall is dominant because it has the capital T. And so what you will see is that 100% of the offspring will be tall. So just make sure when you're analyzing things, if a question asks for genotype, you're giving the alleles. If it's asking for phenotype, you're giving the physical expression of the trait. So in this case, a cross between a homozygous dominant plant and a heterozygous recessive plant will lead to 100% heterozygous offspring. So it's impossible to have a homozygous dominant or a homozygous recessive offspring from this cross. And in terms of phenotypes, all of the offspring will be tall, so it is impossible to have a short offspring arise from this cross. So I want to give you a second example of a monohybrid cross, and this is still looking at the trait of height, but this time the tall plant has a different genotype. So this time the tall plant is a heterozygous, but we are crossing that with a homozygous recessive with a short plant. So again, first step in a Punnett square is to identify the genotype of the two parents, and in this case it has been given to you. So you have a heterozygous plant crossed with a homozygous recessive. Second thing is to determine the two types of genotypes that each plant can produce. And for the tall plant, half of the gametes will have the tall allele, half of the gametes will have the short allele. And for the short plant, half of the gametes will have that short allele, and half of the other half of the gametes will have that short allele. Once you've determined the types of gametes the two plants can produce, or the two parents can produce, you fill out the Punnett square. And for a monohybrid, the square is a 4 by 4. And you enter in the alleles found in the gametes. So for this tall plant, tall allele will go across that row, and the short allele will go across the lower row. For the short plant, you fill in the columns. Half the gametes will have the short allele, half the gametes will have the other short allele. And then of course, you just fill in the Punnett square. So the capital T, the dominant allele, goes across the row. The recessive allele goes across the lower row. And then you fill in the columns, recessive allele, and the next column, recessive allele, recessive allele. Once you've filled in the Punnett square, now you analyze. So for the genotypes, notice there are two different genotypes. There's a heterozygous and a homozygous option. And each of them it takes up 2 squares. And remember, each square equals 25%. So for the heterozygous, there is a 50% chance that the offspring will be heterozygous. And for the homozygous, there is a 50% chance that they will be homozygous recessive. So once you've analyzed that, then of course you can analyze the phenotype. So you get the dominant phenotype if there's one copy of that dominant allele. So heterozygotes will be tall. So there's a probability that 50% of the offspring will be tall, and homozygous recessive will be short. So 50% of the offspring have the probability of being short. Now, the one thing I want to point out here is that these are all probabilities. This is not an absolute. So if these two plants, these two parent plants, have only 4 offspring, it is not the case where 2 must be tall and 2 must be short. That is not the case. This is a probability. In this example, it is possible for all 4 to be tall or for all 4 to be short, because it is a probability. Those two parents are capable of producing tall and or short offspring. Now, the way to think about this is gender. Every time a woman becomes pregnant, it's a 50-50 chance whether it's a female or a male. And just because a woman has 4 children doesn't mean that 2 have to be female and 2 have to be male. This is all a probability. So all this analysis here is a probability. One thing I do want to point out, though, is that in this, there is no option for homozygous dominant. So there is no way that a cross between these two plants will result in an offspring that is homozygous dominant. So the Punnett squares can eliminate options, but it cannot give you an absolute number. After going over important vocabulary and how to perform a Punnett square, I now want to go through some of Mendel's experiments and how he came up with his conclusions. So the very first thing Mendel wanted to do is to develop true breeding plants. And to develop a true breeding plant, you have to allow the plants to self-pollinate. And to self-pollinate, that means the flower petals remain closed so that only the pollen from that plant will fertilize the ovules, the eggs of that plant. And the important thing about a true breeding plant is that all of its offspring have exactly the same traits as the parent plant. And if you allow these plants to self-pollinate over several generations, then you can assume that all of the offspring will be homozygous for that particular trait. Now remember the traits that Mendel was investigating. He was looking at seed shape, seed color, flower color, pod shape, pod color, flower position, and height. And so we've gone over some examples of height already, and you know that there are only 2 options for height, the tall and the dwarf. So if he allowed a tall plant to self-pollinate over several generations, then all the offspring of those plants would be homozygous for that tall allele. And if he did the same thing for the dwarf plant, the short plant, allowed it to self-pollinate over several generations, then you can assume all of those plants are homozygous for the short allele. So it was very important that he establish homozygous plants or true breeding plants for each of these characteristics before he performed his experiments. Once Mendel established true breeding plants for each trait, he then decided to crossbreed them. So the true breeding plants were the P generation, or the parents. And in this case, the trait is flower color. So he had a true breeding purple plant and a true breeding white plant. Now, in order to cross them, he had to prevent self-fertilization. So to prevent that, he removed the stamens from, in this case, the purple flower. Then he wanted to breed the purple flower with the white flower. So what he had to do is use a brush to pick up pollen from the white flower and then fertilize the purple flower. And then the resulting seeds were grown into plants, and this would be the F1 generation. So these are the two parents, the purple flower and the white flower. And of course, these resulting seeds would grow into the filial one, the next generation. When he did this, he noticed something, a recurrent pattern. So in this case, you have stem length. So we've talked about tall and short before. So these are the parents. This is the tall parent, so homozygous dominant. This is the short parent, homozygous recessive. So this is the parental generation. And what he noticed is when he crossed them, all of the F1 had the dominant phenotype. So it's as if the recessive phenotype disappeared in the F1 generation. But then what he did is he allowed the F1 generation to cross-pollinate. So basically like mating or crossing an F1 with an F1. And that gave rise to the F2 generation. And what he noticed with this is that the recessive phenotype reappeared. And he did this many different times with many different traits. So he did stem length, he did pod shape, seed color, flower position, flower color, and pod color. So he started with true breeding parents, a dominant parent and a recessive parent. And no matter the trait, what he noticed is in F1, it showed only dominant phenotypes. But when he allowed the F1s to self-pollinate, or basically an F1 by F1 cross, in the F2s, he got almost exactly the same results. That 1/4 of all the experiments, 1/4 of the offspring showed the recessive phenotype. So the ultimate results he got was a ratio of 3:1. 3 out of 4 showed the dominant phenotype, 1 out of 4 showed the recessive phenotype. These observations led Mendel to develop the law of segregation. The law of segregation basically states that the two alleles in the parent segregate or separate from each other during gamete formation so that only one allele is transmitted to the offspring. So basically what he saw is in his P generation, he had his two true breeding parents, so the homozygous dominant for the tall plant and the homozygous recessive for the short plant. And each of those plants contributed only one allele to the offspring. So then the F1 generation, they were all heterozygous. But because the tall allele is dominant, they all appeared tall. But when he took that F1 generation and allowed it to self-pollinate or to breed with itself, then the gametes that are produced, and this is where the Punnett Square comes in, the gametes would be half dominant allele, contain the dominant allele, half would contain the recessive allele for the eggs, half of the sperm would contain the dominant allele, half the recessive allele. And if you go through the Punnett Square, then 25% probability that, or the probability that 25% of the offspring would be homozygous dominant, probability that 50% of the offspring would be heterozygous, and that about 25% of the offspring would be homozygous recessive. And in terms of phenotype, what you would see is that probability-wise, 75% or 3 out of 4 would be tall, and 25%, 1 out of 4 would be short. And that is the data that he acquired over all of those experiments. So that told him that first of all, the alleles segregate during gamete formation so that only one allele is given to each offspring from each parent. And also it supported his particulate theory of inheritance because the allele, the phenotype is short, seemed to disappear in the F1 generation, but then it reappeared in the F2 generation. So that told him that that short allele was unchanged. It was just hidden by the dominant allele in that second generation. Even though Mendel lived about 200 years ago, his use of the scientific method and the mathematical analysis he did on his results led him to make conclusions that modern genetics supports today. So the two basic conclusions are the particulate theory of inheritance. So the idea that there are physical alleles that are transmitted from parent to offspring and give rise to the traits, and also his law of segregation. So those are supported by modern genetics. And just to show you that they are supported by modern genetics, those alleles are basically genes. So the alleles, the genes, they are located on the chromosomes. And to remind you of the chromosomes, you have two sets of chromosomes, one set from your mother, one set from your father. So these are the homologous chromosomes. They have the same size, same centromere location, and they carry genes for the same trait. But they can be different alleles. So that is supporting the idea of the particulate theory of inheritance. Now, in terms of the law of segregation, this relates to meiosis. So before the cell divides by meiosis, those two chromosomes are duplicated. And remember, each chromosome is going to be composed of two sister chromatids. And because they're duplicates, they're genetically identical, each sister chromatid has the same allele. So the maternal chromosome will be duplicated and the paternal chromosome will be duplicated. And each sister chromatid of the paternal chromosome will have the same allele. Then going through the process of meiosis. So remember meiosis, the whole purpose is to take one cell that is diploid, 2N, and produce 4 haploid gametes. So they have N chromosomes, they're haploid. And meiosis involves 2 rounds of cell division. So in the original cell that is diploid, you have the chromosome made up of 2 sister chromatids, and these 2 red chromosomes have the dominant allele, and the blue chromosomes have the recessive allele. And after the process of meiosis, each gamete is going to have only 1 chromatid. And these 2 gametes will have the dominant chromatid. These 2 gametes will have the recessive chromatid. And so that's the law of segregation. During gamete formation, each gamete is going to contain only 1 allele. When analyzing his data from the monohybrid crosses, Mendel determined that the 2 alleles for a trait separate during gamete formation, and this led him to develop the law of segregation. He then wondered what would happen to the alleles in a dihybrid cross. Di means 2, so dihybrid cross is looking at the inheritance of 2 separate traits. And for this example, it could be seed color. Seed color could be one trait where yellow is dominant and green is recessive, and the other trait could be seed texture. Seed texture, where round and smooth is dominated and wrinkled is recessive. So those would be two different traits, and Mendel wanted to see what happens to the alleles during that cross. And he came up with two hypotheses. First hypothesis is dependent assortment. And the idea of dependent assortment is that certain alleles sort together. So the simplest way to think about this is maybe the two dominant alleles will travel together to the same gamete, and then the recessive alleles would travel together to a different gamete. So that's dependent assortment. The other hypothesis is independent assortment. And with independent assortment, it's completely random which seed color allele ends up in the gamete with whichever seed texture allele. So these were the two hypotheses that he came up with. If dependent assortment is true, then that means that the dominant alleles would always travel together. They would always be found in the same gametes, and then the recessive alleles would always be found in the same gametes. And if you do the prediction, run the Punnett Square with this, the ratio you would get would be a 3-to-1 in your F2 generation. If, on the other hand, the alleles sort independently, then you would get all possible combinations in the gametes. So sometimes the dominant gametes would go together. Sometimes you'd have a yellow gamete with a yellow allele with a wrinkled allele, or a green allele with a round allele, and sometimes the recessive alleles would go together. And you'd get the same thing for the other parent, the other F1 parent. And if this was the case, the ratio you would end up with would be 9-to-3 to 3-to-1. So 9 out of 16 would have both dominant phenotypes. 1 out of 16 would have both recessive phenotypes. And then the two ratios in the middle would have 1 dominant phenotype, 1 recessive phenotype. So that was the prediction. So for dependent assortment, the phenotypic ratio would have been 3-to-1. If it's really independent assortment, then the phenotypic ratio would be 9-to-3 to 3-to-1. And when Mendel performed many experiments testing this and then analyzed all the data, he found that the data looked similar to this, and the phenotypic ratio was approximately 9-to-3 to 3-to-1. So that proved or supported the idea of independent assortment, and repeated experiments that he did led him to develop the law of independent assortment. Now that I've mentioned a dihybrid cross, I just want to give you a practice problem so you can do a Punnett square on a dihybrid cross. So if the question here is, what are the results from a cross between a short plant that is heterozygous for flower color and a plant that is heterozygous for height and has white flowers? So using the steps of the Punnett square, the very first thing to do is to figure out the genotypes of the parent. So the first parent is a short plant. So short is the recessive allele that is heterozygous for flower color. And in pea plants, the two colors are purple and white. Purple is dominant, white is recessive. So usually use the letter of the dominant trait. So capital P is purple, and white, the white allele is lowercase p. So that's the first parent. So figuring out the genotype. Second parent is a plant that is heterozygous for height. Heterozygous for height, one tall allele, one short allele, and has white flowers. To have white flowers, that's a recessive allele, so two recessive alleles. That's a lowercase p. All right, so first thing, find out the genotypes of the parents. Second thing, find out the type of alleles that the parents can produce. And so this is where you can use FOIL, first outside, inside last, or you can just sort of look at it. The way I look at it is in every gamete, you're going to have one T and one P. So for this first parent, this T is going to go with this P half the time. So you're going to have this lowercase t and the capital P. Then half the time, this T is going to go with that little p. So you'll have another gamete with that lowercase p. Then this T will go with the dominant P. And then this T will also go with the lowercase p. All right, so those are the 4 gametes that can be produced by that parent. And then this parent, the gametes that can be produced, this T will go, the capital T will go with that lowercase p some of the time. Some of the time it will go with the other lowercase p. And then the lowercase t will go with that p. And it will go with this p part of the time. Okay. All right, so those are the potential gametes. Now, in a dihybrid cross, the Punnett square is a 4x4. So it is a 4x4. Because each parent can produce a maximum of 4 different types of gametes. So we'll take the gametes for this parent and put them over here. So you have a short allele with a purple allele, uppercase purple allele. You have a short allele with a white allele. You have a short allele with a purple allele. And you have a short allele with a white allele. And then for this parent, you have a tall allele with a white allele. You have a tall allele with a white allele. And you have a short allele with a white allele, short allele with a white allele. And once you've figured out the gametes, then you just fill the dihybrid cross, or the dihybrid Punnett square. So if there's a dominant allele, uppercase, that always goes first. So we fill in this column first. And just do this column too, because there is a dominant T allele. And then this is lowercase. So short alleles, short alleles, short alleles. So all those T's are filled in for the column, and then it go across the rows. So lowercase here, and throughout the whole Punnett square, because all the rows have short alleles. And then do the same thing for the purple allele. So purple, this is dominant, so it's easy to do the capital P first. And this capital P first, to make sure that the capital P is the first one if it is present. And then fill in the recessive P's. Recessive P, so all the rows are filled in. And then fill in the columns, and those are all recessive P's. Or white alleles here. And it is much easier to keep the T's first and the P's second when you analyze it later. All right, so now it's filled in. Now you need to analyze it. And this can take a little bit of time, but first the different types of genotypes. If you noticed, there are 4 different types of genotypes. So the first genotype is heterozygous for each trait. And I'll change colors. So this is heterozygous. This is heterozygous. I'm going down. This is heterozygous, and this is heterozygous, and there are no more heterozygotes. So this ends up being 4 out of 16, so that is 25%. Then the next one is, I'll just do this a different color. So heterozygous for height and homozygous recessive for color. So this is one, heterozygous for height, homozygous recessive for color, heterozygous for height, heterozygous for height, and that's all. So that again is 4 out of 16, 25%. And then another color. So we'll do the next one, homozygous recessive for height and heterozygous for color. So homozygous recessive and heterozygous for color. That is also 4 out of 16, so that is also 25%. And then the last one is homozygous recessive for everything. Homozygous recessive, so homozygous recessive for both traits. And that is also 4 out of 16, 25%. So that's the analysis for the genotype. And then if you go for phenotype, this would be tall purple plants. So 25% will be tall and purple, 25% will be tall and white, 25% will be short and purple, and 25% will be short and white. And that's how you analyze a dihybrid cross. Back to Mendel. When analyzing his dihybrid data, Mendel concluded that the alleles for two different traits sort independently. So that is his law of independent assortment. So if an organism has two alleles for two traits, so in this case, heterozygous for T, heterozygous for G, then the gametes that can be formed have all possible combinations. So in 25% of the gametes, the two dominant alleles will go together. In 25% of the gametes, the two recessive alleles will go together. And in the other 25% of the gametes, one dominant will go with one recessive. And then the last recessive will go with the last dominant. So that's complete independent assortment of the alleles for those two traits. And this would hold true for a trihybrid cross, and that would involve alleles for 3 different traits, and they would all sort independently with each other. Like the law of segregation, the law of independent assortment is also supported by modern genetics. And we find evidence for this in meiosis. So as you recall, when I talked about meiosis, there is a random lining up of chromosomes during metaphase 1. So in this example, you have a cell that has 2 homologous chromosomes. So this is a pair of homologous chromosomes, and this is a pair of homologous chromosomes. And during meiosis 1, those chromosomes can line up in one way randomly with one maternal chromosome here, one paternal chromosome there, or the other option is that the maternal chromosomes line up on one side and the paternal chromosomes line up on the other. So depending on how they line up, you will get random assortment of the different traits that are on those different chromosomes. And in this example, if you have 3 sets of homologous chromosomes, you end up with 8 possible arrangements in the gametes. And that's all due to how the chromosomes line up during metaphase 1. If a trait is inherited according to Mendel's laws, then there are predictable results when heterozygotes are crossed. And so those are the laws of probability. And again, this relates specifically to genes that follow Mendel's laws. So in a monohybrid cross, when you are crossing 2 heterozygotes, so for only one trait, then the probability is that you will get a 3-to-1 ratio, where 3/4 of the offspring will tend to have the dominant phenotype, and about 1/4 of the offspring will have the recessive phenotype. For a dihybrid cross, when you are crossing 2 heterozygotes with each other, then the probability is 9-to-3-to-3-to-1. So 9 out of 16 will have both dominant phenotypes. 1 out of 16 will have both recessive phenotypes. And then 3 will have one of the dominant phenotypes, one recessive phenotype, and the other will have the reverse. So these are the laws of probability when you cross heterozygotes and the alleles follow Mendel's law. A test cross is a method to determine the genotype of an organism. So in this case, you have a plant that has purple flowers. Purple are dominant, and that means this plant could have the genotype of being a homozygous dominant or a heterozygous. And in this case, you do not know exactly what the genotype is. In order to determine that genotype, you will cross this plant with a homozygous recessive. So the homozygous recessive has the 2 recessive alleles. So when you cross this plant, you have 2 different options. If this plant is homozygous dominant, then all of its gametes will have the dominant allele, the purple allele. So then 100%, all of the offspring will be heterozygous. They will exhibit the dominant phenotype. They will be purple. If instead this plant is heterozygous, then half of the alleles will have the dominant allele, half will have the recessive allele. So when you analyze the offspring, roughly half of the offspring will be purple and the other half will be white. So a test cross is used to determine the genotype of an organism. Many human diseases that are genetic follow the laws of Mendelian inheritance, and a pedigree is one way to visualize or represent the inheritance pattern of a single trait or disease in a family. And in order to understand a pedigree, you have to understand the different symbols and what they mean. So in a pedigree, a square is a male, a circle is a female, and clear symbols are healthy individuals. If they are shaded in, they are affected individuals. And if there is a line across the individual, that means they are deceased. Now with a pedigree, you're looking at different generations, and so the generations have Roman numerals. So the Roman numeral I, number 1, that is the oldest generation, and the subsequent generations are numbered 2, 3, and so on. And parents of a child are denoted by a connecting line, and their children extend down from that connecting line, and the oldest is always to the left, and the youngest child or the subsequent children are to the right. So in reading this pedigree, this is the first generation, so this could be the grandfather and the grandmother, and those are the parents, and these are their children. They first had a daughter, and then a son, and then they had a daughter. And then this daughter had children with this man, so they are the parents of these 2 girls, and then this son had children with this woman, so they had a girl and a boy, and then this daughter had children with that man, so these are their 2 boys. So that's how you represent the people, and then the people who are shaded are the ones with this disorder. So the grandmother had the disorder, her son and her daughter also had the disorder, and then her 2 grandchildren from her son had the disorder, and another grandson from her daughter had the disorder. So that is how you read a pedigree. There are several different types of inheritance patterns for genetic diseases, and one type is called an autosomal dominant. And an autosomal dominant is a gene that causes a disease, and it is found on one of your autosomes. And remember, the autosomes are the chromosomes that number 1 through 22. And if it's a dominant, that means you only need one allele or one gene to have the disease. And this is a pedigree showing the inheritance pattern of an autosomal dominant. And one of the ways to first identify the pattern, the inheritance pattern, is to really start at the youngest generation. And what you want to look for is that any affected individuals Because this is a dominant gene, that means they had to get that dominant gene from one of their parents. So you look at these individuals, so this brother and sister, their father was affected. And then you look at his generation, so this brother and sister, and then actually both their parents were affected. So what you will see with an autosomal dominant is that affected children will have at least one affected parents. And one example of an autosomal dominant disease is Huntington's disease. Huntington's is a mutated gene that changes the shape of the protein, which affects the neurological system. So if one of your parents has a Huntington's disease, that means they have the gene for it, and you have a 50% chance of having inherited that gene from your parent. Another type of inheritance pattern for human genetic disease is called autosomal recessive. And autosomal recessive, again, the genes are found on one of those autosomes, but now it's a recessive allele. So in order to exhibit the disease, you need 2 copies of the affected gene. And this is a pedigree of an autosomal recessive. And again, the best way to identify this is to start at the youngest generation. And here, this pedigree actually has 4 generations. But in this pedigree, you see 2 individuals, a sister and a brother. They are both affected by this disorder. And the first thing you want to do is check their parents. And if you look at their parents, neither parent was affected. So that indicates that this is a recessive disorder. And so both of those parents must have been a carrier of the recessive gene. And then, of course, that affected 2 of their 3 children. So that's how you can pretty much identify an autosomal recessive pattern of inheritance. And one example of this is albinism. So albinism, that's where you lack pigment in your hair, skin, and eyes, and that is a recessive. So if you have 2 parents that are heterozygous, then 25% of their children, probability of 25%, could be albino. On this slide, I will show you different pedigrees so you can practice identifying the inheritance patterns. Okay, always start with the youngest generation, and you look at the affected individuals and determine if they have a parent who is affected. So these 2 siblings have a mother who is affected, that mother was affected, and she has a father who is affected. And you want to check every individual in the pedigree. So if you just go again by the youngest generation, these 2 siblings have a father who is affected, and of course his father was affected, and then that female too also had the same affected father. So because every affected individual has an affected parent, this is an autosomal dominant. In this pedigree, again, start at the youngest generation. This individual is affected, but neither parent is affected. And so, and also you have these 2 affected individuals and neither of their parents is affected. So this looks like an autosomal recessive. A few more practice problems. Again, start at the youngest generation. So this female is affected, but neither parent is affected. And then these 2 siblings are affected, but their parents are not affected. So that looks like an autosomal recessive. This one, you have these 2 siblings who are affected. They have an affected father, and his mother was affected. And you just check everybody in the pedigree. These 2 siblings were affected. Both their parents were affected, and her mother was also affected. So that's an autosomal dominant. And in C, you have these 2 with affected parents, and this female had an affected mother. And on the other side, these 3 siblings were affected, as were both of their parents, as was her mother. So that also is autosomal dominant. And then the last one, you have this affected female. Her father was affected, but you don't have any information on his parents, so you can't make any decisions on that. You have this female who is affected, and then you look at her parents who do not have the disorder, so this is an autosomal recessive. There are many examples where the inheritance of a trait does not follow Mendel's laws. And when that occurs, we refer to it as non-Mendelian inheritance or non-Mendelian genetics. And there are many examples in certain types of flower color that is not inherited by following Mendel's laws. And also eye color is another example of non-Mendelian inheritance. One type of non-Mendelian genetics is called codominance. And this is when there are multiple alleles, and more specifically, multiple dominant alleles. And one example has to do with animal coat color or fur color. In cows, there are two alleles, two dominant alleles, a red allele, which produces a red coat, and a white allele, which produces a white coat. And when you have a cow that is heterozygous for those alleles, then both of them are expressed. There are areas of red coat and white coat, and that's referred to as roan. Humans also have a codominant trait, and that is blood type. And with blood type, there are three alleles. There is a dominant A allele, a dominant B allele, and then a recessive allele. So that's a lowercase I. And the dominant A allele will produce A proteins on red blood cells. The dominant B allele produces B proteins on red blood cells. So if a person is homozygous for A or heterozygous for A in the recessive, they have blood type A because their red blood cells have A protein on them. If they are homozygous for B or heterozygous for the B in the recessive allele, they are blood type B because they have B proteins on their red blood cells. If they are heterozygous for A and B, they express both B proteins and A proteins on their red blood cells, so they are blood type B. If they are homozygous recessive, they have no proteins on their red blood cells, so they are blood type 0 or blood type O. Incomplete dominance is another example of non-Mendelian inheritance. And this is when the heterozygote has a phenotype in between the two homozygotes. And this is often seen in flower color. So in the P generation, you can have red flowers that are homozygous for the dominant red allele. And you can also have true breeding parents that are homozygous for the recessive allele, which produces white flowers. When you cross those two true breeding parents, you generate offspring that are heterozygous, and what you see are pink flowers. And so this looks like blending, but it's not blending because when you cross the heterozygotes and you have the F2 generation, what you end up seeing is the original phenotypes. So the red phenotype will be in about 25% of the offspring. The white phenotype will be in about 25% of the offspring. And still the 50% that are heterozygotes will still have that pink color. So incomplete dominance is when the heterozygote's phenotype is in between the two homozygote phenotypes. Incomplete penetrance is another example of non-Mendelian inheritance. And this is where the dominant allele may not be expressed 100% of the time. And this is often due to environmental factors or other genes that affect the expression. And one example is called polydactyly. And that involves multiple digits on the hands or feet. So this is actually a dominant gene, but it is not always expressed when it's inherited. And it's probably due to some environmental factors or other genes being expressed or not. Pleiotropic effects are another example of non-Mendelian genetics. And a pleiotropic effect is when one gene affects more than one trait. So it's not a direct relationship between like a gene and just flower color. One example is Marfan syndrome, and Marfan syndrome is where a person inherits a gene that affects the development of connective tissue. And this often leads to many other effects. So because of that one gene, you have an effect on the connective tissue, which will affect the skeleton. It'll affect the heart and the blood vessels, the eyes, the lungs, and also the skin. So that's a pleiotropic effect, where one gene has many effects on the body. Epistasis is another example of non-Mendelian genetics, and this is where genes that are not alleles with each other interact with each other. So these might be regulatory genes, or they might be genes that override gene expression. Eye color is an epistatic effect, and there are several genes that actually control whether melanin is produced in the iris or not. So there are several genes that actually lead to the development of eye color. Another example could be hair color. So for instance, you could have genes for brown hair. Brown could be dominant, so homozygous dominant would have brown hair, heterozygous would have brown hair, but homozygous recessive would have blonde hair. So that would be Mendelian genetic inheritance, for example. But if you added to that the gene for albinism, so albinism controls whether melanin is produced or not. So you could have a person who is homozygous dominant for hair color, but if they have the gene for albinism, there won't be any melanin produced in their hair. Also, that would be the same if they're heterozygous. If they have the gene for albinism, they would have no melanin produced in their hair. And the same for if they are homozygous recessive and should normally have blonde hair. So this is an example of epistasis. So epistasis, you have the genes for hair color, but then you have the genes that determine whether the color is produced or not. So even if the person is homozygous dominant for brown hair, if they have genes for albinism, they will not express that trait. Polygenic inheritance is also non-Mendelian, and the name tells you exactly what is happening. Poly means many, genic means gene. This involves many genes affecting one trait. And one of the ways you can identify this type of trait is when you see a bell curve for the trait, for the phenotypes. So what you have are two extremes on two ends, and then you have gradual change from one phenotype to another. And probably most human traits are polygenic. One example is skin color. There are several different types of melanin that affect skin color, and each type of melanin has several alleles. So you have all of those different genes affecting the color of the skin. And what you see is a bell curve of different colors. So going from lighter to a darker color and lots of differences in between. So that indicates that it is a polygenic trait. Multifactorial traits are polygenic traits that have environmental influences. So the phenotypes you see are a result of the combined effect of genes, more than one gene, and the environment. So probably a lot of traits are actually multifactorial traits. One example is coat color in Himalayan rabbits. So the coat color is determined by several types of genes, but also temperature is involved. So in the parts of the rabbits that are cooler, like the ears and the nose and the ends of the legs, those actually trigger the pigment production in that part of the rabbit, so you get the darker color. Allergies in humans are multifactorial. There's probably a genetic predisposition. You inherit a gene that makes you more likely to acquire an allergy, and then being exposed to particular allergens, maybe your age, can affect whether you develop the allergy or not. Another multifactorial trait is cancer. Cancer also has a genetic component, so you can inherit genes like mutations in your tumor suppressor genes or your oncogenes, and then also exposure to mutagens like radiation or chemicals can lead to actual development of cancer. So these are 3 examples of multifactorial traits, and that involves polygenic traits and environmental influences. There are some genetic diseases where the gene is located on the X chromosome, and the pattern of inheritance is a little bit unusual, so it's called X-linked inheritance. So X-linked inheritance relates to the sex chromosomes, 2 types of sex chromosomes, the X and the Y, and females are XX and males are XY. And there are very many differences between the 2 chromosomes. The X chromosome has many, many genes. It's a very large chromosome, has lots of genes, whereas the Y chromosome is very small and has very few genes. And most of those genes that are on the Y chromosome are involved in male sexual development. So for those X-linked disorders, some of them are on this figure. So ocular albinism, muscular dystrophy, androgen insensitivity, SCID, severe combined immunodeficiency, hemophilia, and color blindness. Now the thing about the X-linked disorders is that because males have a Y chromosome instead of 2 X's, whatever allele is on their X chromosome, that allele will be expressed, even if it is a recessive. So we refer to the males as hemizygous, because whatever allele is on the X chromosome, that is the allele that will be expressed. So when looking at pedigrees, X-linked disorders have a particular inheritance, and what you will notice is that in the pedigree it will be mainly males that are affected. So if you look at the whole pedigree and you see that it's mainly males that are affected, that can indicate that it is an X-linked disorder. The most famous X-linked pedigree shows how hemophilia spread throughout the royal houses in Europe. And this begins with Queen Victoria. She was the Queen of England and she married Prince Albert. And apparently she was a carrier of hemophilia because one of her 9 children, one son, Leopold, he ended up having the disorder of hemophilia. So that's how they knew she was a carrier, because it's carried on the X chromosome. Boys inherit it from their mother. And then unfortunately 2 of her daughters ended up being carriers. And both of them had sons, at least one son who had hemophilia, and they both had at least one daughter who was a carrier. And the reason we know their daughters were carriers is because each daughter had at least one son who had hemophilia. So one of the things you want to notice about this is that in this pedigree, all the affected individuals are male. So that indicates that this is X-linked. This is the end of the lecture on Mendelian patterns of inheritance. And in this lecture, I went through Mendel's conclusions from all of his research, how to perform a Punnett Square, and different examples of non-Mendelian patterns of inheritance.